So I'm going to move on to the next speaker, which is Dr. Pat Stieg, which she really does not need any introductions, but I'm going to try my best to give, a very summary of, the work that she has done. And honestly, Pat is, when I do brain metastasis research, she is one of, the seminal people, the leaders of the field, and she is, I would say, as they say, a shero to all of us in many, many ways. So Dr. Pat Stieg is the Deputy Chief of the Women's Malignancy Branch and the Senior Investigator at the NIH and NCI specifically. She was really, and Danny mentioned this earlier, she was one of the first to discover the metastasis suppressor gene, specifically called NME. Her work has been seminal in brain metastasis, specifically with breast cancer. And she is one of those scientists that has, like Saul and many others like us on here, has really taken her work from the bench side and made, and pushed it towards clinical trials and has affected patients. So Pat, really a privilege to introduce you. I think you might still be on mute. No, you're not, but the floor is all yours.
Tell me you can hear me.
Nope, you can hear. Yep, you're good. Go for it.
Okay. Hi, everybody. Thank you so much, Josh. It's been wonderful listening to everybody. I'm going to start a talk about brain metastasis, and my colleague, Carrie Anders, is going to follow with more of a clinical presentation. So I'm going to tell you where we are in the lab, and Carrie can follow up with where we are in the clinic. And you've heard about brain mets from a number of speakers, and of course, Josh works in it. Historically, brain mets were very rare. They occurred very late. Patients were often in hospice. They were just treated with some steroids for edema. And it's now almost 40% of those patients who have metastatic tumors that are either HER2 positive or triple negative. That's an astounding rate. And the clinical course varies. For the HER2 positive patients, brain is many times the first site of relapse after the initial metastatic therapy. And what that means is that systemic sites are either stable or responding. The patient's doing well, she has a life, and this is a very unwelcome thing. In triple negative disease, brain mets often occur in the context of uncontrolled systemic disease. And so that's a bit different. Why we all dread them is they pose severe neurocognitive and physical consequences, particularly both from the lesion and the treatments. And the treatments include surgery to actually take the lesion out, two kinds of radiation therapy. There's something called stereotactic, and what that is radiation just to the lesion. And then there's whole brain, and that is the entire brain in usually 10 fractions. It was originally used if there were, quote, multiple brain metastases, which was like 3. We're now up to 10, or, depending on your oncologist opinion, that we're treating with stereotactic before we go to whole brain. Why? In a percentage of patients, there is a long-term, irretrievable cognitive loss. And it's just awful. Recently in the HER2 space, there are a number of chemotherapy combinations, and I will let Carrie take you through them. Something to remember, once a brain met forms, the patient's at high risk to develop more brain mets. And the other thing I want you to remember is that drugs that have activity, they're FDA approved in the metastatic setting, are often inactive in brain mets, and we want to know why. So I'm going to address 3 different topics in 20 minutes, which is probably impossible, from the lab, where one is still blue sky. We hope to translate it. One is in the clinic, and one I hope will go to the clinic if it keeps looking good. But what we do is you've heard the only way to really know metastasis is to inject tumor cells into a mouse and ask, will they spread? And we have either optimized or made on our own any number of cell lines. I think there's 7 shown here that are what I call brain tropic. If we put them in a heart ventricle, they go all throughout the body, and they definitely grow, make brain metastases. And the first thing you can see is they all look different. And I think that's a good thing. I think they explore some of the heterogeneity that is the human condition. And they're all different kinds and subtypes of breast cancer. And so we try to use 2 or 3 or more of these at any time for any study, and then we start believing it. I won't go into these details, but when we made our first one, we compared it in multiple respects to craniotomy specimens, surgically removed brain mets, and it looked pretty good. So it gives you a little confidence of what you're doing. And so this has been the backbone of much of our research. So the first question I want to talk to you about is, why are so many therapeutics active systemically but not in the brain? And there's going to be multiple answers to this question. Priscilla Brastianos, who's up at Harvard, has shown that there are distinct mutations in brain metastases compared to the patient's own primary tumor. So something else is going on, and that can not only affect the metastasis pattern, but the response to drugs. What I want to show you is heterogeneous and low overall drug levels in the brain metastases. Now, I broke this experiment down into a few slides so that we can all follow along. What we did is we took one of those model systems, we labeled it. It's like a lightning bug. It glows green. We injected it into mice, and we let the brain metastases grow. Once they had grown, we injected the mice with 2 different things. One is just a fluorescent protein. It's like a red lightning bug of 3,000 molecular weight. So this is well above the size of many drugs, and we can argue about antibodies. And we also injected the mice with radioactive paclitaxel. So this is a drug commonly used in breast cancer treatment. We let the drug circulate through the mice, and then under deep anesthesia, we perfuse them. What that means is we push saline into them and get rid of all that drug and all that fluorescent protein in the circulatory system. So the only drug that's left is what has moved into the brain or the brain metastasis. So then we section that brain and see where the mets are and how much drug is in them. Okay, so here is a section of a mouse brain. And remember, I told you green lightning bug was the color of the brain mets. And so this brain is riddled with brain metastases. Okay, now if we don't even take it off the microscope, just switch to the red channel. And now we're looking at the 3,000 molecular weight red protein. Did it get into the metastases? What I want you to see, where is my pointer, is that this big red metastasis over here on the right corresponds to this guy, is highly permeable to that Texas red dextran. This area here, almost no permeability to the drug, and it's a metastasis. So you have everything from high to low drug levels in these metastases in the one animal brain. And if you then go to your radioactive imager for that same section, you see the same trend. You see what lit up with Texas red dextran is very high for uptake of paclitaxel. What was generally negative is negative, okay? So what I think we've said is that the entry of drugs or molecules into brain metastases is heterogeneous. I'm not going to put you through the quantitation, but what we found is that only 10% of the metastases that had the highest permeability, the reddest tumors or the most white on the radioactive scan, showed a response to the drug. In other words, it isn't enough for some drug to get in. A lot of drugs got to get in. You don't need some permeability, you need high permeability. And then I want to ask you, because I know you're going to have an opinion on this, is do we want the whole brain to become highly permeable? I don't think we do. I think this is a recipe for adverse events. You don't want paclitaxel running around your neurons. Okay, Or do we want to selectively increase the permeability to brain metastases? And that's what we're trying to do. People can say, does this occur in humans? This is a pre-surgical study where breast cancer patients coming in for medically indicated craniotomies were given an upfront dose of capecitabine in pre-op, and we looked at the level in their tumor, just like we did in the mice. But here it's a bar graph. If you go to 5-FU, this is the metabolite that can go kill a cancer cell, and each color is a different patient's tumor. And here is the same heterogeneity of drug levels that I showed you in the mice. We did that. We've done the mouse studies for 5 different drugs, same conclusion. We did this for 2 different drugs. So why is it that the brain is so crazy? And it's called the blood-brain barrier. And these are the capillaries that are in your brain. The brain has the most capillaries of any organ in the body. And they don't look like a normal capillary has these pink endothelial cells around it to enclose the blood. And they may be all together or there may be gaps between them. Not so in the brain. The endothelial cells are connected by continuous tight junctions, and they express these pumps. Like if something gets in, they pump it right back out. They are surrounded by pericytes, 2 basement membranes in the feet of astrocytes. Every single one of them provides barrier function such that your brain has a homeostatic environment so like neurons can work. And it's when a tumor forms, Cyrus just showed us, they sit on the outside of these capillaries. But what happens to this structure, this blood-brain barrier, which I'm now calling a blood-tumor barrier? The answer is we don't have a clue. And it's more complicated than that. So here's my blood-brain barrier under the normal brain. And you see them surrounded. These are microglia. These are like innate immune cells that monitor, okay, does everything look good? And they'll just eat anything bad. And neurons, which is what the brain needs to do. Besides the blood-tumor barrier, when the metastatic brain, and you see the tumor cells here on the outside of it, you get what's either called a neuroinflammatory response or a glial scar or astrogliosis. They've got lots of names for this. But what it is that these microglia get huge and angry, and they try to wall off whatever this is that's growing in the brain, as well as the feet of these astrocytes. They get angry too, and also try to wall it off. And this is probably an evolution, a response to, say, traumatic brain injury. I don't think the brain developed this for brain metastases. You see the same thing in traumatic brain injury. They try and wall it off, and some immune cells come in. We don't have a good picture of that.
Okay. Sorry to interrupt you. I don't think your slides are moving forward. We're still stuck on the brain metastasis permeability slide.
You're kidding me.
Yeah.
Okay, let me stop a minute. I am going to.
Why don't you restart your PowerPoint again and just go to those slides?
Okay. Are we having fun yet?
But I have to tell you, you were so clear that it was seamless. So don't worry.
Give me a break. How do I escape? Let me do one other thing very quickly. And that is see if, okay, then let's go back to this. Hello.
It's okay.
Oh yeah, it's just absolutely fine and dandy. Why can't I not pull this Zoom up? God, I love technology. This is where I need a young person.
Or a joke.
Or
you guys have my slides.
I do. Let me see. Let me see if I can pull.
There's Zoom.
There you go. Let me see if I can pull.
Okay, let me see share screen. And wait a minute, I need.
Okay, why don't you go ahead and share your screen again?
I'm so sorry.
No, it's okay.
If you guys have it faster, do it.
Let me see.
Oh, this isn't going to work because I took off my my VPN.
It's okay. Hold on.
Oh my heavens. Okay,
Why don't
you guys are going to have to put it up.
I don't have. It's okay. I don't. Darren, do we have her slides?
Yeah, you do. I sent it.
Yeah, Darren, if you have it, can you post it or can you pull it up?
He texted. He has it.
Yeah, no, I know he has it. Let me see, Pat. Okay, so Darren is going to pull up your slides. Yeah, he's going to.
Does Carrie want to go and we can?
No, here, we have your slides. So Darren, why don't you move forward? There we go. We're going to move forward, and then you just let him know what's.
Can you see them now?
Yep, there we go.
Okay, good. Okay, keep going. And this, this was the uptake in the actual patients. Can I flip it or do you? This was the blood-brain barrier. Next, this was the blood tumor barrier. This was the whole brain. And here's where we are. I'm so sorry. What the lab has found is we have spent 4 years profiling what the blood tumor barrier is. The good news is it's not a chaotic breakdown of the blood-brain barrier. There are consistent differences, 3 different model systems. We see these same differences in human craniotomies. The second good piece of news is we've been able to modulate its permeability just in a model system, but we have changed the permeability of the blood tumor barrier without the blood-brain barrier. We are currently using aspects of the blood tumor barrier to try to deliver drugs. So we have experiments underway that are trying to get more methotrexate into brain metastases using one of these blood tumor barrier systems. And I hope we'll see. This is my blue sky approach. Next slide. Okay, moving to a second project that's ongoing. And this is a lead for the prevention of brain metastases. And those patients who have metastatic disease say, I don't care about that. But I would like to tell you, I think you should. Because as I told you, if a patient gets a single brain metastasis, they're going to get more. It is just time. And so the question is, if you have your first set of brain metastases, can we stop it and not get any more? And that's called secondary prevention. It's a lousy word, but there it is. So next slide. This is the way we have been doing drug studies for forever. We inject the mice, and that bottom green line is after 3 days later, let them recover. We start dosing in drug, and we keep it going. And at the end point, we ask, did they get brain metastases? That is a prevention assay. If that drug stopped brain mets from forming, it's prevention. It is not what we do in clinical trials. What we do in clinical trials is this, and that is you let the brain mets grow, then start the drug and ask, will it shrink or eliminate them? We have now tried 30, maybe 40 compounds. We have 4 with significant preventive activity. We have almost none that have treatment activity. And the drug to everyone's befuddlement that worked the best was temozolomide. Next. Temozolomide is a drug that is A, brain permeable, and B, the primary line of treatment for primary brain tumors, glioblastoma, this stuff. So when we dosed it into mice, what you see here are 4 experiments. And experiment 1, can you see my pointer? Maybe? Okay.
No, you can't. So you just have to describe it.
A clinically relevant dose of temozolomide. In other words, it's something that a patient can achieve. What we found is that no brain mets formed. We went down to 25 in experiment 2, no brain mets. We went down to 5 in experiment 3, no brain mets. And we're down 100-fold before we lost significance. Click, please. And then we did the experiment where we gave the mice either a short course of either temozolomide or vehicle. Temozolomide was early or late. And here you have the survival. The vehicle-treated mice died on time. And you see that both resulted in long-term cures. The let them run around the cage for months on end. And depending early better than late, you had cures. Next slide. There was a lot of additional information. We didn't do 2 mouse experiments and say, let's do a trial. And I won't go through all that for you unless somebody wants to discuss it. And we are aware that there are previous clinical trials. It says temozolomide doesn't shrink and establish brain met in all comers trials. This would be melanoma, lung cancer, breast cancer. It may have efficacy in combination with radiation therapy, and it has 0 systemic activity in breast cancer. Next slide. And despite that, the mice were saying, hey, we're doing pretty well. And Dr. Alex Zimmer, whose picture I had up earlier, has set up this trial long-term, is the phase 2 trial. It's for HER2-positive breast cancer patients who have already had 1 to 10 brain metastases. In other words, it's secondary prevention. Treat it usually with radiation just to the metastasis. We need a systemic agent, that's TDM1. And so it's TDM1 alone or TDM1 with what we call metronomic, that means every day, low dose temozolomide. Next slide. The primary endpoint is not shrinking anything. It's the percentage of patients that do not get another brain met for a year. This is the phase 1. We're in the expansion phase of dose level 3. We have 1 more patient to enroll, and then I will dance across the stage and tell you the data. Next slide. And I'm not going to go into these. I'm terribly over at this point. But we have put a lot of thought into our exploratory endpoints. We are taking CSF and serum. I want to know, like someone previously said, will CSF predict better than what's going on in serum? We're looking at any number of things. Next slide. Okay, the last thing I want to tell you about is sort of in between blue sky and the clinic. And this is the work of Alex Wu, who is one of the most talented postdocs I've ever had. And it addresses a point that you don't hear about much in breast cancer, although Carrie Anders did some of the foundational work on this. And that is, why do patients who are diagnosed with breast cancer at a young age have a poor prognosis? There have been a number of tumor studies that say, yep, poor prognosis, but they have large tumors or triple negative tumors, or they have an African American race, or they have this or they have that. Maybe that's it. But if you do something called multivariate analysis and take every one of those variables out, at the bottom, at the end of the day, a young age diagnosis is an independent predictor of poor outcome. So we went to the tumor databases and went through them with for those mutational events, et cetera, that were present in primary tumors from young patients. And we didn't find much. Few GATA3 mutations. So if it's not the tumor, is it the host? Which was a crazy idea. It has just about killed us. But what we examined in the host is metastasis. And the way we did it, next slide, is to do young and old mice side by side. We took the same tumor cell model system, put it in young mice, old mice. You'd think no difference. Here is the brain metastasis data. You see 4 different model systems. Young, the number of brain metastases in the red bar, old in the blue bar. Every single time, 3 to 4-fold diminution in the older mice. Click. In the 99LN mouse, they also got liver mets. Don't know why. Don't see that in any of the others. No difference in the liver mets. So young women, young mice get a lot of brain mets. There are clinical papers that say young human breast cancer patients get more brain mets. Next slide. Here is a slide where we looked at 4 different model systems of lung metastasis, and I will spare you the trouble. It's the far right-hand column. There's no difference between young and old. So this seems to be a brain metastasis thing. Next slide. I'm going to summarize a whole lot of data for you. When you know there's something going on in a young brain, we ruled out hormonal effects. in our model system, not saying they don't have profound effects. We then went after the immune system, and we found that there are T lymphocytes, neutrophils, granulocytes, microglia in the metastatic mouse brain. I'm not going to bother you with all the data. What we did is we selectively depleted every single one of them, and nothing with the microglia had an effect on the metastatic rate. And if we, in that A box, if we gave mice a CSF1R inhibitor, which will actually kill microglia, it inhibits monocytes and macrophages, it kills microglia, and then injected the model system, it decreased from red to pink in the lower panel, the number of brain metastases. Well, it's interesting because in normal aging, the microglia, as I said, are the native immune cells of the brain. In normal aging, the brain loses microglia, and with it, they gain activated astrocytes. We see the exact same thing in these mice that we've treated with the Plexicon compound, the CSF1R inhibitor. We see reduced microglia, we see activated astrocytes, and reduced brain metastases. So in essence, what I think we're doing, believe me, we've looked at 1 million markers of microglial activation, nothing's changed. We're basically, I think, making these young brains resemble an older brain, which may not sound like the best thing, but if you're facing a life-threatening challenge, it might be a good idea. This CSF1R inhibitor attenuated metastasis, caused the uninvolved brain of the young animal to resemble that of an older animal. And we wonder, could there ever be age-appropriate therapies, or is one size fits all the way we go? We are working with a pharmaceutical company on a what might be an improved version of this. And then we're going to think about how and if to take this to the clinic. Next slide. These are all my collaborators. Thank you so much. I'm so sorry for the foul up. I have no idea what happened. Thank you.
That was great, Pat. And we're okay on timing. And I think it's great that, you're the work that you've done with, the aged mice and showing that correlation with patients, because we see the same. I mean, we all see those observations of the younger, women who do worse. And it's great, that you're leading the field again on figuring that aspect out.
It's killing us. There is, I know people online will know this, there is a young survivor coalition. And I follow their thoughts as carefully as I can. And they're the ones who actually inspired me to go do this.
Yeah, no, we have a lot of inspiration stories. Okay. Mm-hmm. So while Carrie, and we have questions, but we're gonna hold it off until the end. Thanks again, Pat. Okay, so while Carrie gets her slides up, I'm gonna introduce her.